Welcome to section 4.1, Energy and Metabolism. In this section, we'll explore the foundational concepts behind how cells acquire, transform, and use energy. We'll walk through metabolic pathways, energy transformations, and the incredible efficiency of enzymes, all critical to understanding how life functions at the molecular level. By the end of this section, you should be able to describe metabolic pathways, explain the first and second laws of thermodynamics, distinguish kinetic and potential energy, compare endergonic and exergonic reactions, and understand how enzymes work as catalysts. These learning objectives are rooted in your ability to see biology through the lens of energy flow and chemical transformation. All living things require energy to carry out essential functions, from muscle contraction to DNA replication. Energy in biological systems is managed through metabolism, which is the sum of all chemical reactions in a cell. These reactions are not random. They are part of organized sequences called metabolic pathways. Metabolic pathways are step-by-step sequences of chemical reactions, each catalyzed by a specific enzyme. These pathways allow cells to transform energy efficiently. For instance, cellular respiration is a metabolic pathway that extracts energy from glucose, while photosynthesis is a pathway that stores solar energy in sugars. The first law of thermodynamics states that energy cannot be created or destroyed, only transformed. When you eat food, the chemical energy in that food doesn't disappear, it's converted to other forms, like kinetic energy when you move, or thermal energy when you generate heat. The second law tells us that every energy transfer increases entropy, or disorder, in the universe. Think about how your phone heats up as you use it. That's wasted energy in the form of heat. Similarly, in cells, energy transformations release heat, making them inherently inefficient and increasing entropy. Kinetic energy is energy in motion, like a moving molecule or a flying baseball. Potential energy is stored energy, like the energy stored in the bonds of a glucose molecule. Cells constantly convert between these two forms. For example, when ATP is broken down, potential energy is released and used to power cellular work. In exergonic reactions, energy is released, like in cellular respiration, where glucose is broken down to release energy. In endergonic reactions, energy is absorbed. Photosynthesis is a great example. It takes in sunlight to build glucose. Cells rely on both types of reactions to survive. Enzymes are biological catalysts. They speed up reactions by lowering the activation energy required. Importantly, enzymes are not used up during the reaction. They can be reused. Each enzyme is highly specific, usually catalyzing just one kind of chemical reaction. Initially, enzyme function was described by the lock and key model, where the substrate fits the enzyme perfectly. We now use the induced fit model, which recognizes that enzymes slightly change shape to better bind the substrate. This dynamic interaction makes the process more efficient and accurate. Enzyme activity depends on conditions like temperature and pH. Some enzymes need cofactors, non-protein helpers, to function. Enzyme inhibitors can block activity, which is one way cells regulate metabolism. We'll look at two common types of inhibitors next. Competitive inhibitors resemble the enzyme's normal substrate and compete for the active site. Their effect can often be reduced by increasing substrate concentration. Non-competitive inhibitors bind elsewhere on the enzyme, changing its shape and preventing proper function, regardless of how much substrate is present. Allosteric regulation is when a molecule binds at a site other than the active site. This can either activate or inhibit the enzyme. It's a key strategy cells use to regulate their metabolic pathways quickly and efficiently. Feedback inhibition is a type of allosteric regulation. Here, the final product of a pathway inhibits an earlier enzyme in the sequence. This stops the cell from wasting energy and resources by producing more product than needed. It's a built in control mechanism. Many medications work by inhibiting enzymes. Ibuprofen blocks enzymes involved in pain and inflammation. Antibiotics inhibit enzymes in bacterial cells. Researchers are also studying enzyme inhibitors for treating cancer and metabolic disorders, highlighting how this topic connects to real world healthcare. Energy and metabolism are the foundation of life at the molecular level. From the laws of thermodynamics to enzyme regulation, understanding these processes helps us appreciate how cells are finely tuned to use energy efficiently. As you continue studying biology, remember that energy transformations and enzyme action are recurring themes in nearly every system you'll encounter.